Hi, my name is Laura Schaerer. I am a PhD student in the Department of Biological Sciences at Michigan Tech. Today I'll be talking about my research titled Microbial Communities of Bilgewater, Boat Surfaces, and Portwater, a Global Comparison. Each year, thousands of boats travel the world's oceans carrying everything from coffee to automobiles. Many of these ships have tanks which are used to carry ballast water to stabilize the boat and to increase safety and stability. Many organisms have traveled in ballast water, which has resulted in the introduction of invasive species. Bacteria are ubiquitous in nearly every environment on Earth. In addition to ballast water, microorganisms may live on other areas of a boat, although shipping regulations have recently changed to be mindful of microorganisms in ballast water. Little is known about bacterial hitchhikers on other parts of the boat. My research aims to understand how microbes from portwater may impact the microbial community on a boat's hull and bilge. In the largest boat microbiome study to date, we sampled boats and water in 20 ports on 3 continents to characterize the hull and bilge microbial communities on boats. Figure 1 demonstrates that there is a diverse microbial community living on the hull and in the bilge. Our principal coordinates analysis shows that the water and boat samples are similar in terms of microbial community composition since they cluster tightly together. Figure 2 compares differentially abundant organisms between the water and the boat. In general, more bacterial species are enriched in the water than on the boat, but few species are present on the boat that are not also present in the water. Cyanobacteria are phototrophic organisms which need sunlight to grow. Figure 3 shows a diverse community of cyanobacteria is present in the bilge, which suggests that outside water gets into the bilge, since these organisms would be unlikely to thrive in a dark environment. Figure 4 shows an abundance of taxa closely related to pathogens in each of the sample types. This shows that the hull and bilge may be home to pathogens which have potential to travel with the boat. Figure 5 summarizes our results from Source Tracker. Source Tracker is a program which learns what the microbial community from a set of sources looks like. The program is then able to classify the proportion of each source contributed to another set of samples. This model reported that the water microbial community sourced 40% of the bilge microbial community and 52% of the hull microbial community. In conclusion, our findings show that microorganisms from the water impact the boat microbial community. In the future, we would like to explore the impact of terrestrial and airborne sources on the boat and measure the persistence of location-specific microbial signatures on the hull and in the bilge of the boat. Thank you for listening. If you have any questions, feel free to email me. My email address is at the bottom of your screen, or check out our publication in Applied Environmental Microbiology.